Welcome back, and we are now going to complete part 2 of chapter 4, cell structure. So when we left off, we had been talking a lot about the cytosol and cytoskeleton and those parts of the cell, and now we start moving in towards what we more often than not consider the actual organelles of the cell and the intracellular structures. So we start with what's known as the endomembrane system. And the endomembrane system consists of the nuclear envelope, the endoplasmic reticulum, both the rough ER and the smooth ER, the Golgi apparatus, and lysosomes. Also, peroxisomes are sometimes considered part of the endomembrane system, but they're really actually what we call a semi-autonomous organelle, and we'll get to those towards the end. But the endomembrane system itself is kind of this big network, and all of these different parts, they all are separate and they have separate functions and structure, but they all work together to carry out the larger processes of the cell. And one thing they do all have in common is they are all part of the protein synthesis pathway. One thing to remember about our cells is that proteins are what do all of the work. Yes, we have DNA, we have this genetic material that determines everything, but proteins are the real workers. They're the ones who actually make things happen. And proteins, in order to make things happen and make them happen correctly and in a timely manner, our cells have to produce them, and they have to synthesize them. They have to process these proteins. Sometimes the protein needs a sugar attached to it or a lipid molecule, something like that. So the endomembrane system is a big part of the protein synthesis and processing that goes on in the cell for cellular function. The plasma membrane itself, what we would call the cell membrane, is also considered part of the membrane system, but since it is on the outside of the cell, it's not really part of the endomembrane system. And the endomembrane system all of the different parts of it are oftentimes directly connected to each other or indirectly. With indirect connection, they are passing molecules and other compounds back and forth through vesicles. So we start with the nuclear envelope. The nuclear envelope is actually a double membrane, whereas we think of phospholipid bilayer in our cell membrane. The nuclear envelope is actually a double membrane, so there's 2 layers of phospholipids there. The outer membrane will be continuous with the membrane that forms the endoplasmic reticulum, in particular the rough ER, the rough endoplasmic reticulum. The inner membrane, also composed of phospholipids, really is just used structurally to help with protecting the genetic material in the chromosome. The inner membrane is lined with lamin proteins, and these lamin proteins form what is referred to as the nuclear lamina. The nuclear lamina helps to keep the nucleus in this kind of 3-dimensional structure, and it also allows for some anchoring of some of the microtubules and actin filaments used for chromosomal territories. Embedded throughout the entire nuclear envelope are nuclear pores, and nuclear pores are very, very important structures. They have specialized proteins that surround the actual pore itself. They're not just holes, but they're proteins surrounding them that regulate the passage of mRNA out of the cell, as well as regulating passage of anything coming into the nucleus. So not only is our cell membrane semi-permeable, or what we call selectively permeable, but the nuclear envelope is also selectively permeable. And with good reason. Our genetic material is inside of this nucleus, so we need to make sure that it is very well protected. Now, all of the material that's found inside of the nucleus, of course, is not considered part of the endomembrane system. So here we have just kind of a bigger to smaller type of picture here. We have, of course, here's our nucleus in the cell itself, right? And we have our genetic material. That's all this wiggly kind of red stuff here. And we have our nucleolus. The nucleolus is a region within the actual nucleus itself. Here's a transmission electron micrograph, kind of showing you where we have the nucleolus, and there's chromatin, which is our genetic material wrapped around histone proteins. Here's the nuclear lamina just lining the inside here. All this squiggly stuff outside is actually endoplasmic reticulum. The nuclear envelope is just on the outside of that structural lamina, like they're showing you here. You can see here they're also showing you the two membranes. This is a scanning electron micrograph on the outside of the nucleus showing you these nuclear pores. So you can see these nuclear pores, there are just millions of them. The nucleus is covered with them. And if we get even smaller here, we get to the actual rendition here of the nuclear pore. And you can see that this pore complex here is going to highly regulate, sorry about that, it's going to highly regulate what can actually move in and out of that nucleus. And that's, again, really important because we need to really protect our DNA. The cell only gets one copy of that, so it needs to be protected. Now, the nucleus itself in its entirety is the largest organelle in the cell. It's usually visible upon staining with a light microscope. It contains a couple of different parts. We, of course, have our chromosomes. Now, a chromosome is our DNA, and the DNA is wrapped around these protein subunits called histones. So the DNA molecule itself is not the chromosome. It's actually the DNA wrapped around its histone. So structurally, a chromosome is made up of a combination of DNA and histone proteins referred to as chromatin. The nuclear matrix is composed of nuclear lamina, and an internal matrix is connected to it. This is used to create what are called chromosome territories. Our chromosomes don't overlap each other. So up here in this little picture here with all these different colors, here's a colored micrograph. This was done probably with Photoshop. They took an electron micrograph and used coloring on it. But over here, they've taken those colors and they've given each of the colors a number, and each number corresponds with, I did it again, each number corresponds with one of the chromosomes. Now remember, we have two of each chromosome. So I want you to see here we have right here in this light blue is chromosome 9, and over here is chromosome 9, right? So we have two copies of each chromosome, and notice none of them overlap. We have no overlapping. Here's 17 in the dark green, and here's 17 in a red as well. So we have, here's the X chromosome, so this is a female cell. Right? So we have we have one X. I can't see a Y. Oh, we have a Y, so this is a male cell, excuse me. So here's our Y chromosome. The X and Y chromosomes are the sex chromosomes, and are the only ones that don't match. All right, so then we also have in our nucleus, we have the nucleolus. The nucleolus is most easily seen in cells that are in a non-dividing state. If the cell's in a dividing state, then we're not really capable of seeing the nucleolus. And the only thing that's really important, well, it's not the only thing, but what is important about the nucleolus is that it's a very specific region within the nucleus in which ribosomes are made. And this is important because remember, ribosomes come together to help translate mRNA, and mRNA is also made in the nucleus. So we need to make sure that ribosomes and mRNA leave the nucleus separately. We don't want the ribosome making a bunch of proteins inside of the nucleus that could possibly be harmful to other material inside of it. So the whole function of the nucleus, its first and foremost function is to protect our genetic material. It has to be protected from the outside environment. It's used to organize chromatin because our genes that determine who we are located on our chromatin, and we need that chromatin to be accessible, and our transcription factors, the proteins that start the transcription and kind of use our genetic material, have to be able to find what it is that they're supposed to be transcribing. And regulating the expression of our genetic material is also occurring in the nucleus. Ribosome assembly in the nucleolus, both the large and small subunit are formed. We also find the ribosomal RNA and RNA proteins in there. And it takes place in the nucleolus because it's so close to the genes that are required for encoding them. So nucleolus is where ribosomes are made. But those ribosomes, just so you know, those ribosomes will not become functional until they enter into the cytoplasm. Now, the endoplasmic reticulum is our next stop in the endomembrane system, and it is actually divided into two regions. So the endoplasmic reticulum is really quite large. It's incredibly large, not quite as big as the nucleus, but it kind of wraps itself around the nucleus. And it is formed out of these flat fluid-like tubules, and they're all kind of smashed up together. They're sometimes referred to as cisternae, and the ER membrane is actually continuous, if you remember, with that outer membrane layer of the nuclear membrane. It encloses a single compartment called the endoplasmic reticulum lumen. Lumen just means inside of something, the inside. Now, the two regions of the ER are the rough ER, referred to the RER, and the smooth ER, and referred to as the SER. Now, the rough ER is rough because that flat kind of tube-like membrane that's all smashed up together is covered in ribosomes. So these ribosomes leave the nucleus and they go out into the cytoplasm, but not all protein synthesis takes place in the cytoplasm. Most of it actually takes place in the membrane of the rough endoplasmic reticulum. So a ribosome and mRNA will come together, and after they come together, the ribosome will embed itself into the endoplasmic reticulum membrane. So the membrane itself looks kind of rough. It has all these little studs or dots all over it. All of those dots are ribosomes. Now, once a ribosome embeds into the ER membrane, it will carry out its protein synthesis, and when it's done, it will actually leave the membrane. So although the rough ER is kind of covered in all of these ribosomes, they are not permanently attached. They kind of come and go as needed. The rough ER is responsible for initiating protein synthesis, not all proteins, but most of them. It also takes part in the processing and sorting of proteins. We make hundreds of thousands of different proteins in our cells, and all of those proteins need to be processed, and they all need to be determined where they're supposed to be going in order to carry out their function. The rough ER is responsible for preparing new proteins for the membrane to carry them into the membrane. So any of our proteins that are what we call membrane embedded, this would be for both organelles and our cell membrane. And most importantly, the rough ER begins the process of what's called glycosylation. In glycosylation, sugar molecules will be attached to lipids or to proteins. So glycosylation is a very important process because the sugar molecules, specific sequences of sugar molecules, are often used by our cells for recognition to each other and to our immune system. From there, we move further away from the nucleus, still part of the endoplasmic reticulum, but we no longer have ribosomes embedding, and proteins are moving through the lumen of the endoplasmic reticulum from the rough ER to the smooth ER. And the smooth ER, there's no ribosomes, and a primary function of the smooth ER is actually detoxification. You see this a lot in liver cells. We produce glycogen in the smooth ER, which is carbohydrate metabolism. We use the smooth ER in heart cells and cardiac cells to maintain a calcium balance for our heart to beat and some of the electrical signals that our heart uses. And then, of course, if we're going to create or modify any kind of lipid, that's going to occur in the smooth ER as well. So the smooth ER is smaller than the rough ER, but as far as size goes, but it is really very important in some of the different functions of our cells. For example, in detoxification of liver cells, if you've ever heard of the term cirrhosis of the liver, that just simply means inflammation of the liver. And what's happening in cirrhosis of the liver is it's very common among lifelong alcoholics. And people who drink lots of alcohol for a long period of time, their liver starts to compensate for this constant bombardment of alcohol. Alcohol detoxification takes place in the liver. So all of this alcohol keeps coming in. So the liver cells say, well, I gotta keep cleaning up all of this. So I'm just going to make more endoplasmic reticulum. And the size of the smooth endoplasmic reticulum starts to overcome the cell. The cell begins to keep on creating more and more smooth ER. It has less ER. The cell gets very crowded. And this starts to throw everything off balance. When it throws everything off balance, the cells now turn towards inflammatory signals. The immune system is responding. It's just not a very good thing. So these cells get really big. And if you ever get the chance to look at liver cells from a lifelong alcoholic or someone who has cirrhosis of the liver for some other reason, take a look at see if you can find the smooth ER. It will be very large in those cells compared to others. The rough endoplasmic reticulum and the smooth endoplasmic reticulum are continuous with each other. So here on the left we have kind of our cartoon drawing, and you can see these yellow dots here are the ribosomes all studded within. And this is, you can see in here, this is the lumen. Here's the nuclear envelope, and it's continuous here with the rough ER, and then the rough ER comes out here to the smooth ER. These little guys right here are vesicles. They are coming off of the smooth ER, probably going to the Golgi. Here's another picture kind of showing the same thing. Here's our nuclear envelope. We're going in the other direction here. Nuclear envelope, and here are the nuclear pores. And it's continuous. They're showing you the continuity here with this rough ER. And the rough ER is really big, but eventually as we get away from the nucleus, we start to lose the ribosomes, and we enter over here into the smooth ER. So that concludes part 2 of chapter 4, and we're now going to, in part 3, we'll talk about the remaining organelles and some differences between plant and animal cells.